Well, good afternoon, everyone. Hopefully everybody can hear my voice. I am Bernie Rossoff, and as chair of this study, I'd like to welcome everyone to this first open meeting for the assessment of NIH research on autoimmune diseases. This is a congressionally mandated study that is sponsored by the National Institutes of Health. I'm joined this afternoon by the committee members who are all participating virtually. I'll ask them to introduce themselves shortly. Before we get further along, I want to note that this is an open, on-the-record session. Interested individuals have been invited to attend as observers via web conference. I also would like to remind everyone that this is an information gathering session. It's important for everybody to recognize that this is really an information gathering session for the committee, and of course for others who will be in attendance. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, please understand that the committee has made no conclusions at this time. The committee has made no conclusions at this time. Comments made by individuals, including invited speakers and members of the committee, should not be interpreted as positions of the committee or of the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a very rigorous review by experts who are anonymous to the committee. And the committee must then respond to this review with appropriate revisions that adequately satisfy the Academy's report review committee and the National Academy of Science president before it is considered an official Academy's report. So this is a rather rigorous process so that the report has finished and completed all those individual processes. I'd like to explain the format of our meeting today. We will start off by hearing the official charge to the committee from Dr. Ellen Goldmans of the National Institute of Allergy and Infectious Disease. And that will be then followed by Alex Keenan, who is with the Senate Committee on Appropriations, who will provide general context regarding Congress motivations for requesting the study and perspectives on how Congress might use this committee's report. We will also hear from Dr. James McNamara from the Division of Allergy, Immunology, and Transplantation at the NIAID, who will present an overview of NIH structure as it pertains to funding and research for autoimmune disease. Then we will have two framing presentations. Ms. Virginia Ladd of the American Autoimmune Related Diseases Association will share an overview of the evolution of and the current state of autoimmune diseases. And Dr. Jane Buckner from the Benaroya Institute will present the state of research on autoimmune diseases, including progress made to date and opportunities for the future. After each of the remarks, the committee will be able to ask brief clarifying questions. All attendees can indicate their questions or comments at any time during the event by typing them into the Q&A box at the bottom of your screen. That should work quite well, and we hope to have all who you would like to ask a question actually do that. If not, we have a process to allow those questions to be submitted at a later time. The Academy study team will be tracking comments for speakers and the committee. Now, reporters who would like to talk with members of the committee are kindly asked to touch base with the staff officer, Henrietta Awo Osai-Anto, by emailing autoimmunestudy.nas.edu. That's autoimmunestudy@nas.edu. Once again, let me remind everybody that this meeting is being recorded. Now, I'd like to ask the committee members to introduce themselves, giving their names as listed on the attached roster in alphabetical order. We will do that, and brief bios of all committee members are available for everybody online. My name is Bernie Rossoff. I'm a gastroenterologist, currently professor of medicine at the Zucker School of Medicine at Northwell Health, and a member of the board of directors of Huntington Hospital Northwell Health System, and also the board of overseers of Northwell Health System in New York. Glinda?
I can't activate my camera. Okay, got it. Hi, I'm Glenda Cooper, and I'm an epidemiologist. I'm currently in a different role. I'm the Director of Science and Research at the Innocence Project, focusing on forensic science and wrongful conviction and criminal justice reforms. But I do have a long history of research in epidemiology of the systemic autoimmune diseases and looking at environmental risk factors for diseases.
Deirdre?
I'll also need my camera, my video ability. Okay.
There you go.
Thank you. Hi, everyone. I'm Deidre Cruz. I'm a professor of medicine in the Division of Nephrology at Johns Hopkins, and also am Associate Director for Research Development for the Johns Hopkins Center for Health Equity.
William?
Yeah, I'm Bill Duncan. I'm the Vice Provost of Research at East Tennessee State University. I have a background in drug development in a variety of diseases, include autoimmune diseases.
Sonia?
I am Sonia Friedman. I'm an associate professor of medicine at Harvard Medical School at Brigham and Women's Hospital in Boston, and I'm director of women's health at the Center for Crohn's and Colitis, and I've been treating patients with Crohn's and ulcerative colitis for 25 years.
Lisa?
Hi, I can't get my camera to start either. It's been stopped by the host.
Click on the video icon and that should do it.
Yeah, no, it didn't do it.
No, it didn't do it.
It says it's stopped by the host.
Here you go.
Okay, but anyway,
We have you now.
excuse me?
We have you now.
Oh, okay. Thank you. My name is Lisa Iasoni. I'm professor of medicine at Harvard Medical School. I'm based at the Center for Health Policy Research at the Massachusetts General Hospital, and I've been investigating healthcare disparities for people with disability for more than 20 years.
Andrea?
Just waiting for my video. Okay, there we go. All right, my name is Andrea Knight. I'm a pediatric rheumatologist at the Hospital for Sick Children in Toronto. I am also an assistant professor of pediatrics at the University of Toronto, and I have a clinical research interest in childhood onset lupus, particularly neuropsychiatric aspects, as well as overall mental health for rheumatology patients. Thank you.
Michael?
Okay, waiting here for the video. Okay. There. Okay. My video is on. I don't know if I'm
We have you.
visible.
Yes, we
Okay.
have.
I'm Michael Lockshin. I'm professor of medicine and obstetrics gynecology at Weill Cornell Medicine and head of the Barbara Volker Center for Women and Autoimmune Diseases at the Hospital for Special Surgery. In the early to mid 90s, I was extramural director, then acting director of NIAMS.
Jill?
Hello, I'm Jill Norris. I'm professor and chair of the Department of Epidemiology in the Colorado School of Public Health. My research interest is in the development of autoimmunity and the progression to disease with regard to type 1 diabetes, celiac disease, rheumatoid arthritis, and lupus.
Emily?
Hello, everyone. I'm Emily Summers. I'm an associate professor at the University of Michigan. I'm trained as an epidemiologist with a primary appointment in the Division of Rheumatology. I also have joint appointments in environmental health sciences and OBGYN. I have a longstanding history in autoimmunity and autoimmune diseases, including the connections between and clustering between autoimmune diseases, and also a specialty in lupus. Thank you.
As you can see, we have quite a list of experts on the committee. You've heard their brief bio introductions, and all willing to learn from the experiences we're going to have today by members who are going to speak to us for the remainder of the day. I'd like Awo to introduce the team from the National Academy. Awo.
Hello, good afternoon, everyone, and thank you for joining this meeting. My name is Awo Sayanto, and I am the study director for this particular meeting, for this particular, study. We also have two other team members on this call, Kristen White and Layla Maymond, who are supporting the study, as well as Rose Martinez, who is the board director for the Board on Public Health at the National Academy's Health and Medicine Division, and who is the advisor on the study as well.
So thank you, Awo. And I must make the point I made yesterday that the team is really responsible for putting this together in such a wonderful fashion. And as you will see, as we go through the day, the process was well crafted, and everybody has been well charged with their responsibilities for today and for the remainder of the time this committee will meet. So let me start with the sponsor charge to the committee, which will be given by Dr. Ellen Goldmunz. Dr. Goldmunz is the section chief for the Rheumatologic Autoimmune Disease section at the NIH, National Institute of Allergy and Infectious Disease, and a practicing rheumatologist. Everybody, please welcome her as she presents the charge to our committee. Dr. Goldmunz.
Thank you very much. So I don't have to take care of my own introduction. Just to clarify one thing, I'm actually trained as a pediatric rheumatologist. Now is the video on?
Yes.
Okay, sorry about that. It was flashing little signals at me. So I'm trained as a pediatric rheumatologist, not an adult rheumatologist. I've been at the NIH for about 17 years at this point. And as task leader for the NIH contract with NASEM, I want to thank you for inviting me to deliver the charge to the committee today. Can I have the next slide, please? So in fiscal year 2020, the Senate appropriations language included direction to the NIH as a whole to contract with NASEM on several studies, one of which was focused on autoimmune disease conditions. Most often, at least in my experience, the congressional language is directed to a particular institute or center, but in this case, the language was directed to the NIH as a whole. The congressional language was preceded by a statement of congressional interest in autoimmune disease research, and the summary of their interest and the background information for their directive is summarized on this slide. The language noted that autoimmune diseases affect approximately 5 to 7% of people in the US. Some are more common in women than in men. Many women are affected during their childbearing years, and many of those also live with a second autoimmune disease or comorbid condition. Despite the prevalence and impact of these diseases and conditions, there's no single office within the NIH tasked with coordinating research across the agency or examining the complex interplay among these diseases and conditions. Can I have the next slide, please? So my next slide lists the highlights of the congressional directive that was given to the NIH, which ultimately served as the basis for the statement of task with NASEM. The list of topics is quite broad and is summarized here. The NIH was asked to contract with NASEM to review NIH's research efforts in autoimmune disease, an area of predominantly women's health. Specifically, the review needs to evaluate NIH's research in autoimmune disease and coexisting disorders, epidemiologic trends of autoimmune disease in the US population, and barriers to diagnosis. The congressional language also directed that the report explore barriers to research and promising areas for future research. Finally, the NASEM report was to include recommendations on how the NIH could improve coordination of activities and research into these diseases. May I have the next slide, please? So last spring, in order to meet the requirements mandated by Congress, NIAID was tasked by the NIH OD to develop a contract with NASEM, and it was entitled the Congressionally Mandated Assessment of NIH Research on Autoimmune Diseases. The contract is of 18 months duration. I am the task leader, and I have 2 people helping me as alternate NIH task leaders, both of whom you've been introduced to. One is Dr. Lisa Begg, who is a Senior Research Program Officer in the Office of Research on Women's Health, and Ms. Susan Cooper, who is the Director, the Office of Program Planning, Operations, and Scientific Information at NIAID. Next slide, please. With that as introduction, I'd like to present to you the charge of the committee as written by the NIH and accepted by NASEM. Next slide, please. So these are the specific tasks for the committee to address. We have defined 8 tasks, which are outlined on the next 3 slides. The first is to assess both NIH-sponsored and other research to identify epidemiologic trends in autoimmune diseases. For common autoimmune diseases, including those that are overrepresented in women, to evaluate the NIH research portfolio with particular attention to issues such as risk factors, diagnostic tools, barriers to diagnosis, treatment, prevention, and prospect for cure. We also have tasked the committee to review NIH's research activities related to specific autoimmune diseases, the occurrence of multiple autoimmune diseases in individuals, and the interplay of autoimmune diseases and comorbidities. Can I have the next slide, please? The list of tasks is continued on this slide. The committee is tasked with assessing trends in NIH research. Do they reflect the changes in epidemiology, the availability of new research tools and technologies, and emerging concepts and knowledge? The committee is tasked with identifying barriers to NIH-sponsored research and research gaps for autoimmune disease, and to identify promising areas for future NIH-sponsored research for autoimmune disease. May I have the next slide, please? These last 2 tasks are on this final slide, almost final slide, penultimate slide. The committee is specifically asked to evaluate institute and center structure in support of NIH autoimmune disease research. In particular, are institute programs and collaborations addressing important needs, and are there opportunities to enhance coordination? And finally, we've asked that NASEM produce and publish a final consensus committee report summarizing the committee's findings, conclusions, and recommendations, including NIH accomplishments, challenges, and possible solutions to the challenges. May I have my next slide, please? So I want to say thank you for participating in this effort. We understand that these projects require a substantial commitment on the part of members of the committee, as well as the NASEM staff. We're very aware of the significant time you will spend, and know that in the end, the report will be of high quality. We look forward to reading your analysis and receiving your recommendations. And with that, I'd be happy to answer any questions.
So thank you very much, Dr. Goldmans. If committee members do have questions, please use the raise hands feature in the participants tab at the bottom of the screen, and either I or Awo will call on you to speak. Also, please, before you do ask your questions, please identify yourself for the record. I do have one question that perhaps I can start with. In some of the discussions we had yesterday, we noted that back in 2015, there was a downward trend in autoimmune disease funding. I think, and also perhaps in some of the applications for autoimmune disease research. $968 million is estimated for 2021. The numbers don't seem to be changing very much. So what is there a big change in the status of grant applications that you're aware of? Anything that we should know? Have the applications remained the same? Have they changed over time? And if so, how or how much?
So Dr. McNamara is going to talk to you about an overview of NIH funding, and he'll show you some graphics. So I don't want to steal his thunder.
Okay.
So I think if you have more questions after that, we can come back to it. How about that?
Okay, that's great. That's perfect.
Okay, Dr. Lakshan, please unmute yourself and ask your question.
Okay, hi, Ellen. I have two generic questions. The first is, how in fact do you define autoimmune disease? And I bring this up in the context of genetic illnesses, infection triggered, including things like the COVID and autoantibody sort of things. That's the first part of the question. The second is that diseases tend to be thought of in very short-term intervals. You do studies that will be 2 years or maybe 5 years or something, but the autoimmune diseases with which we deal are lifelong, and they change their patterns, they change their mechanisms, and they change they vary on damage accrual as well. And so I wanted to ask both questions about your definition and what the boundaries of autoimmunity are for the purposes of this study.
So I don't know that my personal definition is as important as sort of the common concept. I think we think of autoimmune diseases as a chronic condition. I don't think at this moment I would name post-infectious autoimmunity as an autoimmune disease unless it becomes chronic. And in the case of COVID, which is not the point here, I don't think we've demonstrated that yet. And I think that the answer is very complex, and I don't think that Congress defined exactly what they were interested in when they were talking about autoimmune disease. These are long-term diseases. They do change over time. And Mike, I guess I'm not 100% sure what you're asking me with the second question. Yes, they do change over time. Yes, there are different phases of diseases, and research into all of that is important.
Yeah, you more or less answered. One of the things is that I'm looking at, this committee has the freedom to go explore all of the variants, not just specific named diseases, not just lupus, not just MS, and so on, but to be quite broad in its definition.
I think you can be quite broad, but I think in terms of using the NIH portfolio and the public resources available to you, you will find it easier with the more common, I mean, there will be more data on the more common illnesses. Just relative to NIH funding and research activities.
Okay, thank you.
Emily?
Sorry. Okay. So I'll follow on the last point. It dovetails into my comment.
Would just introduce yourself, that would be helpful.
I'm sorry. Sorry again. I'm Emily Summers at the University of Michigan, epidemiologist. So my question relates to how you're using the term common. On your slide, you had that distinction made for one of the bullets. And with, so first I'm wondering if that's just meant as a functional term to aid in feasibility, because as you just mentioned, there would be more data available, presumably for the more common of the diseases. But just a point I would like to make is that even some of the well-recognized autoimmune diseases such as lupus hover right around that definition of rare disease that can be used by the FDA or other groups. Of course, definitions also vary by country in terms of what is defined as common. So I just wanted a little clarification on how that term is intended in this context.
Yeah, it's not meant as a precise definition of 1% or 2% of the population, if that's what you're getting at. Certainly lupus is holds an awful lot of interest and is near and dear to many of our hearts and causes an awful lot of serious illness, disability across a long a lifetime and across many different ethnic groups. So yes, we're not asking for only those of greater than 1% in the population. Does that answer your question?
Yes, thank you.
Oh, Lisa?
Thank you. I appreciate your going over the charge to the committee again. And you have a very nice kind of list of things such as barriers to diagnosis and all the way up to cure. And I will self-identify as somebody who's had multiple sclerosis for 44 years. And so the concept of cure is actually not relevant to me. And I'm wondering whether you're interested, whether part of the charge to the committee could go beyond the notion of cure and talking about research about living with conditions that frankly are not immediate prospects for cure.
So I think that in terms of, it's a big charge. You know, and you have 18 months, and this should, I think, be data-based. So yes, I mean, the list of tasks that I gave you were examples. So we didn't talk about prevention either there. So cure, prevention, healing, you know, these are important topics. And I guess the question is, what is NIH doing and how have they moved as research has evolved? And have we kept up with what we need to keep up with? So I don't know if I can actually answer your question, but you're going to see, I think, what comes out of it. But cure, yes, would be wonderful. You know, the reintroduction of tolerance is, of course, a holy grail. Prevention may be another thing we want to think about. I didn't put it on the list. It wasn't specifically in the congressional mandate. Does that help you?
Yes.
You
Thank
don't
you.
look satisfied.
I will say that this is going to be an evolving process, probably of understanding the scope of where we should go with the notion of these diseases not being curable and the research that might be conducted to think about the experience and the adaptations and technologies that can assist people to live with incurable disease and disabling disease.
Thank you. I do not see any more hands raised. Oh, there is one. Andrea?
Yes. Hi, Dr. Goldsmith. From the pediatric perspective and thinking about the autoimmune diseases that span the lifetime, the charge, as I understand it, didn't explicitly call out children as affected individuals. I just wanted your comment because I do think, obviously, from my perspective, and also I think, you know, the perspective of the disease across a lifetime, it would be important to consider children. I just wanted to clarify that.
So absolutely consider children. And I mean, as a pediatric rheumatologist, of course, children have been my primary, at least clinical interest for a long time. But also some of the autoimmune diseases are, after all, very common and have their onset in childhood, particularly something like type 1 diabetes. So I think absolutely children should be included in this. And yeah, does that help you?
Yes, thank you.
Seeing no more hands raised, thank you very much, Dr. Goldmans. I really appreciate your comments. And if I heard correctly, we certainly will include children in our charge. And in terms of the question asked earlier in terms of cure and prevention and healing, that also would be part of the research that we would consider in our deliberations. So we appreciate your comments and thank you.
I actually did have one additional question. I'm sorry, I had some difficulty with the hand raising.
Go right ahead, Dee. Go right ahead.
Okay, thank you. And Deidre Cruz, I'm a nephrologist at Johns Hopkins. My question was regarding whether an examination of the actual workforce or the investigators themselves who are conducting this work would be a part of your what you would view as a part of the scope for our committee. And so by that meaning, you know, who is conducting the characteristics of the people who are entering this field of research.
So I think this is a little bit dependent on how you. Well, let me back up here. This is a report supposed to be based on NIH research activities, and clearly NIH can't do its work unless it has an adequate and representative workforce. So to that extent, and we do significant training and fund training all through the spectrum of medical and research trainees. And if we're failing in that, we may be failing in our NIH research mission. So while I really don't think that it was meant to be a workforce question, and I can't really speak to what Congress had in mind, and maybe Alex Keenan can give you a better idea of that. I just have the congressional language that you also have, which did not mention workforce. But I suppose if you see a great failing in the workforce, that impacts what NIH can do in terms of NIH research on autoimmunity, then it's something that might be considered. I don't want to say yes or no, but certainly if you don't have the workforce, you can't do the job.
Thank you for that. Any other questions? Once again, thank you very much. Much appreciated.
You're very welcome.
We'll move on to the study background given by Alex Keenan. Alex is he's with the Subcommittee on Labor, Health, and Human Services, Education, and Related Agencies at the Senate Committee on Appropriations. He will provide some background on policymakers' motivation for mandating this study and how Congress may use the committee's report, all of which are important to us as we go through our process and deliberations. Alex?
Good afternoon. I was able to catch the last few minutes of the discussion, which was really interesting. And it sounds like people are going in a lot of, I think in general, good directions. You're right that the directive in the statement of managers is fairly limited, as these things usually are. But I think if there's anything that sort of you can fit within those last 4 sentences of the language, which is fairly broad, then you certainly have the ability to sort of look at that issue. Where this language sort of originates is over the last few years, I'm sure it's probably been longer, having lots of constituents, having lots of advocates come in to see Senator Murray, and seeing this pattern of, frankly, since we're not scientists up on the Hill for the most part, seeing this pattern of constituents and advocates who may mention a particular illness or condition and then group it sort of broadly as an autoimmune disease. And frequently, of course, they would have more than one autoimmune disease. And being aware of this report from a decade ago that the National Academies had produced, which has sort of identified this as a particular area of concern, Senator Murray wanted to sort of see what things look like a decade later. Had progress been made? And I think one of the factors that also played into this is the nature of NIH's organizational structure, being an agency of 27 different institutes and centers, which is really quite unique in the federal government. I can't think of anything else like it. And you really have, in some ways, 27 different agencies trying at times to be one agency, but they're not. And things tend to fall through the cracks. So it's not unusual. We've seen at times with some conditions for one institute to say, well, that's really not ours. That really belongs over there. And then have another institute say, well, no, that's not ours either. And so some concern in particular, I think, with these conditions of could they be falling through the cracks? And then also just a history, which I think the agency is certainly sensitive to and continues to grapple with, of certain groups falling through the cracks as well. That we continue to see this with, say, COVID research, where it's been difficult to get sort of a representative mix of people into clinical trials. So with all those things in mind, that's where this direction and the funding came to conduct this study to see where is NIH 10 years later? Has it made progress? Are there still barriers? Do you have things falling through the cracks? It could be issues that have come up that were mentioned in the last few minutes of, does it have the right workforce to conduct this research? When you're looking at sort of promising areas of research, certainly it could be cures, it could be diagnosis, it could be prevention. Where are the opportunities, and is NIH pursuing them? And I think related to that, at some point, and I can't remember, I'm sure at one point I knew, the Office of Women's Health Research was created. If you were to look at this year's House report, they would be noting with concern that funding for that office has not grown. So even though NIH funding has been growing now consistently for the last 5 years, that office has remained by and large flat. So I think what you see is some concern that, with patterns like that, and how then, of course, it rolls into down into individual diseases and conditions. And so that is one of the reasons also at the end, one of the questions is, if there are barriers, what are the recommendations that you all would have in terms of fixing them? And it could include the sort of radical idea of maybe creating a new entity, which hopefully would not be needed. But if that is perceived to be sort of the only answer, then of course, Senator Murray and other members of Congress would be interested in having that viewpoint.
That's very helpful. Very helpful in terms of commentary of questions that we raised yesterday, Alex Keenan. I think that a look back is always good. It really stimulates looking at where there have been deficiencies. If you remember, we did that with Error is Human and Crossing the Quality Chasm 15 years after that report came out. And we found clearly that not much was done that was recommended in the first report. And subsequently to revealing that, a lot of activity has occurred in the area of quality and performance improvement. So hopefully the same will be with our report going forward. So as a reminder, if committee members have questions, please raise your hands and in the participant tab, and I'll acknowledge your questions. Michael?
Hello, I'm Michael Loxton. I'm just a little curious. The way you presented that is asking the way NIH is doing this, but one of the things that I learned when I was at NIH is that oftentimes what was spent and what were the directives came from Congress, that there were specific requests from sometimes a group of people in Congress and sometimes just single individuals that an institute do this or do that. Is there a way of getting an overall picture as opposed to one-on-one requests from individual congressmen or senators?
I'm not sure I understand the question. If you're talking about NIH's, are you talking about NIH's budget?
Yes.
I mean, the agency has an unusual degree of control over its budget. Because it is a scientific agency, you do see some money it does have that sort of directive nature that you're speaking about. And I would think sort of the most well-known right now would be Alzheimer's research. But by and large, the vast majority of funding that NIH gets is really at the, it's at the discretion of the IC directors in terms of how that money is used. So, I think of all sort of the agencies and bureaucracies I've worked with, and I really can't think of another agency that has that kind of trust and flexibility.
Good, thank you.
Seeing no other questions, thank you very much for this. Really appreciate your
My pleasure.
perspectives. And if any questions do arise, we'll be sure to communicate with you subsequently. Thanks once again.
Great, thank you.
We will now go to a little overview of the NIH institutes and centers and funding structure. Dr. James McNamara will provide that. He is the Chief for Autoimmunity and Mucosal Immunological Branch in the Division of Allergy, Immunology, and Transplantation at NIAID. So please welcome him as he presents a brief overview of the NIH ICs as it pertains to autoimmune funding and research. Dr. McNamara.
Hi, this is Jim McNamara. Thanks for welcoming me here this morning. Can you see me? Is my video working?
Yep.
Okay, thanks. Okay, so why don't we go to the next slide? So mainly I'm going to be talking about the funding, sort of different categories related to this for autoimmune disease research in the next few slides here. And the tool that I'll be predominantly using is what's called here RCDC, Research, Condition, and Disease Categorization. It's a system that the Institute uses for identifying the funds that are associated with different autoimmune diseases and many other types of diseases. As a bit of history here, this was started in 2008, and you can see in the first bullet here that this was as a result of a congressional mandate in 2006 that NIH developed a more comprehensive and transparent public reporting system. So prior to that time, each institute and center sort of did their own thing for identifying their different funding streams and how they were being used. Whereas from this point onward in 2008, there was the RCDC was implemented, and that improved consistency and transparency. It also recognizes that these are the official NIH spending figures for each one of these different categories, such as the autoimmune diseases I'm going to be talking about. And it's going to be through 2019, which is in this last bullet, but that's because that's considered actual money. So going back from 2008 to 2019, those are actual figures that are associated with these disorders, and they're estimated for 20 and 21. So slides that I'm going to be showing you are typically from 2008 to 2019. Nope, not ready for that yet. Thank you. It's a computerized reporting process, and that means that in order to give this consistency to the evaluation of these different dollars that are allocated to these disorders, there is a data mining sort of process that goes on that's related to specific words and phrases of words that were set up by a number of different sort of scientific experts in these areas within the NIH that have been sort of validated for what they're pulling forward. So this fingerprint that is identified for an individual disease has been developed over time to give this consistency. There are currently 292 publicly reported categories. That has grown substantially over the past decade, and there probably will be more that will be added in the future. But currently, we're looking at 292 reported categories. I wanted to just take one step back to say this is all part of what's called REPORT, the Research Portfolio Online Reporting Tool. So this is one of the funding tools within that overall report system. And there's a lot of other valuable information there about individual grants and how they break out, descriptions of them that can be used there. The entire RCDC portfolio that you'll be hearing a little bit about from me is also something that is fully downloadable and searchable and analyzable by others as well. Okay, why don't we go now to the next slide? So in talking about these 292 different categories, one of those is the autoimmune diseases parent category. So parent is sort of talking about a higher level category. And then there are children that are subcategories within that group. And I've listed here both the parent autoimmune diseases and then the children that are identified underneath that. So for 2019, the total category is $988 million. And then you can see how that breaks out for these other subcategories, including inflammatory bowel disease, lupus, MS, etc. So those are the ones that are limited to this group, and all other sort of autoimmune disease research rolls up to make up the rest of that $988 million figure. Okay, why don't we go to the next slide? These are just a couple of comments about this. The RCDC does not prorate research and grants and contracts. So a couple points there. Vast majority of these are grants. You'll see that there are some contracts, but it does include grants and contracts. It does include intramural and extramural, as we'll see too. And overall, what this is trying to convey is that if there is a grant that is that has aims that are related to both lupus as well as an aim that's related to MS, those are both included in those different categories funds. The overall roll-up of the cost, though, it was still a single grant with a single budget figure, and that is what gets rolled up into the total. But there's, so that if you look at the sub-children here, the subcategories, the children of these, they don't necessarily add up to the total number because of they're represented in both areas. Stating sort of the obvious based upon the previous slide, there are many well-known autoimmune diseases that do not have their own category. So there's some big categories like that: type 1 diabetes, Sjogren's syndrome, celiac disease, just to mention a few. We know that there are many different autoimmune diseases. That sort of varies by who the author of that is, but it's well over 80. It's well over 100 different autoimmune diseases. So it's only a few that are individually identified within RCDC. Okay, why don't we go to the next one? So then this is looking at the funding over the time period that we have the actual figures for. So 2008 through 2019. It shows in red the NIH overall budget. That's on the axis to the right versus the autoimmune disease funding, which is in blue, and the axis is on the left. Overall, I think we could take a couple of things from this. There's autoimmune diseases has basically paralleled the NIH funding, and during that time period, and just looking at the figures that we're looking at here, the NIH funding has gone up about 30% over that time period. And there's been a similar increase in autoimmune diseases over that time period as well. Okay, next slide. So there are different ways that we could sort of look at breakdowns of these of this funding, different ways to slice and dice this. I'm going to give a couple of ways here. This is sort of one of the more obvious ways to look at this, and this is how the breakdown is by institute. And you can see how that plays out across the axis here as each institute is represented. There's been, I think, using this tool, the RCDC, has been really a great advance in trying to have people talk about whether we're talking apples and oranges here and talking about this over the past more than a decade, but it's not perfect. And I can draw your attention to the bar graph at the end. That's RMAP and TID. That's for roadmap and type 1 diabetes. And as an example, we know from that there have been specific congressional funding of type 1 diabetes in the special diabetes program. And in 2019, which this is representative of in this slide, that was $100 million and $150 million in type 1D special diabetes program money, and that isn't represented in the overall money that's captured here in the RCDC. So although there have been, this has been a great tool for us to look at for individual institutes, it's not without these faults, and as part of the work of this committee, you may want to look into that a bit more, but I wanted to just outline that for you. And then looking at the next slide, this is another way to sort of break down this data, and that's to look at the different funding mechanisms. So here we're looking at extramural funding mechanisms, and as you can see in the top bullet, extramural support in for autoimmune diseases and RCDC is roughly 86% of the total. And the breakdown here, I think, is what you might anticipate. Of course, the single largest mechanism type was the R01. Those that I think probably most people listening to this or know that that is the standard sort of workhorse grant mechanism. The R, for those that don't follow this as closely, the R stands for research. There are other mechanisms that we have for, of course, and that includes the U's and the P's, also standing for program project grants, multi-project grants, center awards are typical P's. The U's are cooperative agreements, frequently multi-project to some extent as well. And so they sort of fall into a somewhat different category. Then there's the career and training grants. And then the sort of leftover bucket of all other types of R's besides the workhorses of the R01s, which are sort of single project grants. R21s are more exploratory, smaller grants. And other grant types or contracts. Other grant types, you know, sort of thinking what that might represent. Off the top of my head, I can think of one, and that's using, I think what are called X grants, and that's sort of a intramural, extramural sort of a collaboration type of grant. So there are rare grants, as you can see under the other types, that fall out of the traditional R's, P's, U's, and the training and career grants. I mentioned that the RCDC does include grants and contracts, but under autoimmune diseases, there are only a few rare contracts, as you can see. But this does represent sort of the breakdown of all of these grants. I'm sorry, all of the funding that does go into the autoimmune disease sort of a family of grants and contract support. Okay, and this is the last of my slides. Another way to look at how this money is broken down is to look at solicited research versus unsolicited research. So the majority of the work is, of course, unsolicited research, and that's typically in the R's and some U's. But there's also solicited research, which is more where NIH drives some of those research activities to address specific scientific gaps or specific needs here. The example that's given here is clinical trial networks as a fairly obvious one where we work together on different projects. But I wanted to sort of bring this to your attention that there is these different buckets of solicited versus unsolicited, because that first bar graph that I showed you sort of emphasized sort of silos, because it's looking at the funding that is happening for each one of the, it's distributed to each one of the institutes. But actually, when we're talking about solicited research, there is the opportunity for much more sort of collaborative processes that occur across institutes, and in fact does work that way. So I wanted to just bring this other type of way of looking at research to your attention. Looking at how that breaks down into the different ways I was able to use RCDC to show you how the funding follows that. It's more difficult for solicited and unsolicited research. So I don't have those numbers for you. That's something that could be pulled out, but it's not part of the more readily available figures that are available in RCDC for me to be able to break that out for you today. And I think that's, can I look at the next slide? There is no next slide. Just thanks for your attention and in listening to this presentation.
Thanks very much, Dr. McNamara. Once again, if committee members have questions, please use the raised hands in the participants, and we will be happy to call upon you. Deirdre?
Thank you. So thank you for that overview. I just had a question around whether there's data available on the partnerships between NIH and other agencies in terms of funding some of these awards. So for example, an NIH PCORI sponsored study on an autoimmune disease. Do you have a sense of how broad that type of portfolio is?
No, you're correct to identify that those types of collaborations do occur across a variety of different sort of relationships. The one that has, I think, received a lot of interest recently was the Accelerating Medicines Partnership, which was sort of across NIH, but also looking at industry partners and funding that. But there are others where there are foundation support for grants too. It isn't something that we can easily pull out of RCDC to be able to to quantitate that better for you. Those types of things are annotated in Reporter, which is the tool that you can use to look at these. So it's possible to kind of pull that out, but it would be a more laborious thing than what is already sort of pre-populated within RCDC for as the tool for looking at this. So it is possible, but it's not easily attainable for me to give you a simple answer to that question.
Thank you.
I only have time for the people who are already in the queue, so we'll go to Bill, Glinda, and Michael. Bill? Not hearing from.
I hit it and then it back off. Okay, sorry about that. Dr. McNamara, thank you for your overview. That was outstanding. Clearly, there's a large number of institutes and centers involved in supporting research on the wide array of autoimmune diseases. Yeah, what mechanisms does NIH use to try to coordinate activities across campus, the NIH campus, and a variety of diseases, and also to ensure that other areas that may not be receiving as much attention as they should be can be targeted?
Mm-hmm. Yes, thanks for that question. I would say there's two sort of generic ways that I can respond to that. One is that all of the solicited research, for example, it goes through a process of review by all of the institute. So if NIDDK is putting out a request for applications, that is something that by process for approval throughout the NIH, all of the other institutes see what that proposal is and has an opportunity to interact with NIDDK with that, and also has the opportunities to join some of those types of proposals. So it's sort of built into the system within the NIH that for each one of these types of proposals that goes through, that there is a process of vetting across the NIH different institutes, and allows for the opportunity for different institutes to weigh in on that proposal. There's also another element to this, which is separate from that, and you also may be referring to, and that's the Autoimmune Diseases Coordinating Committee, which is a process that allows the different committees the different institutes to talk about within the committee, as well as in sort of some aspects of that are also open to the public, for there to be a discussion about plans in a general sense for that are moving forward as well. Because of the nature of the process of having these programs move forward, there's a a certain element that needs to be confidential until these RFAs and RFPs become publicly available at one time, to not seem to overly favor one group of people that have access to information to over another. So there needs to be sort of a well-timed rollout of these types of programs. But those are the two main ways I would say that the internal mechanisms that the NIH has, as well as the Autoimmune Disease Coordinating Committee, where there's an opportunity to discuss those programs more broadly as well.
Thank you. Glinda?
Thanks. I just had, I think, 3 clarifying questions. One is, did you say that the intramural research programs are or are not included in the totals that you were given? The second, just, so did you say if multiple diseases are addressed by a single grant, it's counted in each of the children, but it's not, the amount isn't, double counted or triple counted for the total at the top? Is that the way that works? And then my third one is, can you explain, or did you explain and I missed it, why some of these diseases don't have their own child categories within the parent of autoimmune diseases?
So thanks for those questions. I'll try to jot down some notes as you were going. Yes, but to begin with, intramural was included in that 988 for 2019. It was 86% extramural. Second, yes, you did understand correctly what I said, that there is not double counting in the overall numbers, as I understand it. Although there is sort of identification of that grant towards both of those diseases if it was lupus and MS, as I was giving as an example. But I'm going to step back on both those points to say that the RCDC and report is really out of the NIH OD, and I am not an expert in that area. There are others that are more expert in this than I am. And if further information about RCDC and the inner workings of that would be helpful to the committee, I'd urge you to have sort of a separate sort of discussion with the people that work on this. And then you came to the final question, which is sort of related to part of this answer that I gave before, that what about other children of the autoimmune diseases? Why aren't there 30 or 40 or more? And some of that is that this was originated from Congress, and there was sort of the original groups that came forward, where it was a smaller group of these. It is a fair amount of work to identify these fingerprints and to make sure that they're accurate and how they impact other fingerprints for diseases. But we can add some. We have, for example, recently gone to the staff at RCDC to request that one of these categories be available for celiac disease. And so that goes into a queue. They have a list of other diseases that they work with, and it does take time and effort to kind of do that. So those can be expanded to the event that's possible.
Okay, thanks.
Thank you. Last question, Lisa.
Thank you, Dr. McNamara, for this very clear presentation. I have a question about what the implications are of this kind of decentralized structure for study sections and for the expertise on study sections for reviewing grants that might be related to autoimmune disorders.
Okay, I don't think I'm going to be able to answer that question very well. There may be other people at NIH that can do that, because I was mainly focusing on the funding part, which is really a separate issue from the grant review and study sections themselves. And I think there were probably others that can better answer that than I can. I'm sorry.
So Lisa, we'll park that and try to submit it for further answer. Thank you very much, Dr. McNamara. This was really very helpful to the committee. I'm sure we'll have other questions as time goes along, and you may hear from us sometime in the near future. And thanks very, very, very much.
Happy to help in any way I can.
Thank you. The next session will be the state of autoimmune disease in the United States. Very broad topic, and to tackle that is Virginia Ladd. Ms. Ladd is the founding president of the American Autoimmune Related Diseases Association, an organization that she founded to bring a national focus to autoimmunity and also to increase collaboration in autoimmune research, in autoimmune education, awareness, and advocacy. She is currently president emerita, and she serves as an advisor to the president of AARDA. Welcome, Virginia. We're looking forward to your comments.
Thank you. Thank you very much for the opportunity to speak to this group. Next slide, please. This is some of the background facts that just we'll touch on. Some of it has already been discussed, so I probably won't spend too much time on that. But the history of autoimmune disease is somewhat of relatively new to the world of research, because it was just 70 years ago that it was recognized that the immune system could produce antibodies against oneself. Then research took another approximately 40 years before it became really more robust and more of interest. And then when you look at most research really has been within a single autoimmune disease. So that's the kind of the history. ARDA is a national voluntary health organization. We were founded 30 years ago. I actually am the founder, and it was founded, as was stated, to foster collaboration in research, advocacy, public awareness, and patient services, and also to have autoimmune disease recognized as a disease category, particularly also that autoimmune diseases run in families. And one of the projects of ARDA has been to also found the National Coalition of Autoimmune Patient Groups, which brings together 39 groups that represent singular disease, autoimmune diseases, or groups of autoimmune diseases. There's over 130 autoimmune diseases, and that changes periodically up and down. 75% are women, and in some diseases as high as 10 to 1. Exceptions to that would be type 1 diabetes and Crohn's disease. The sex bias of autoimmunity has attracted enormous attention in research. However, it still remains unsolved. Another fact of autoimmune disease, autoimmune disease is one of the top 10 causes of death in women and children under the age of 65. Some autoimmune diseases are more prevalent in minority communities, and in diseases such as lupus, the disease may be more severe with a higher death rate. So some of the contributing factors, which will be discussed later and more in depth in this presentation, are environment, genetics, and infection, and how they interplay. Lack of awareness and funding. ARDA conducts a study every 5 years to track public awareness of autoimmune diseases. And the last study indicated that only 29% of Americans can name an autoimmune disease. Why that's important is because when asked a family history, if a patient has an autoimmune disease or they're seeking a diagnosis, they really don't know to offer the information that another family member has a different autoimmune disease, which might be the clue that provokes looking into that possibility. So that is an issue that needs much more awareness, and I think would really help with the diagnosis, because 67%, I believe it is, of patients with autoimmune disease have a member of their family with another autoimmune disease. So that's important information. Regarding funding, NIH has not really increased much over the last 5 years, although we heard today that it stands at $988 million. It took a little bit of a dip in 2018, but out of the 100 autoimmune diseases supported by NIH, approximately 42% of NIH budget was allocated to just 9 autoimmune diseases. And that might be where the most robust research is that would impact on others, but nevertheless, it is a fact. And there are many autoimmune diseases that have a significant impact on patients, but really have very little research. So autoimmune diseases can affect any part of the body. can be localized, and can be systemic. Autoimmune patients can develop more than one and often several autoimmune diseases over their lifetime. I myself will disclose I'm an autoimmune patient. I was diagnosed with lupus when I was 23. I'm in my 70s now, and I've collected 5 autoimmune diseases. Just recently, MS on top of lupus. So over a lifetime, the autoimmune patient is susceptible to developing more complicated autoimmune problems. Next slide, please. This is kind of a schematic which shows the target for autoimmune diseases. And as you can see, multi-specialists would be involved in treatment. So because a single autoimmune disease may be systemic, the patient often has to see more than one specialist. And there are several issues regarding coordinated care regarding that issue. Next slide, please. So these are some of the issues major issues that we at ARDA have identified. And some of the issues that I will address some of these issues I'll address separately. We did hold a meeting of our Scientific Advisory Board, which is an international board of prominent researchers. And these also are issues that were brought up by the members at our meeting we held last week to gather the data. Next slide. So there is a significant need for current and accurate data to make informed decisions for the millions impacted by autoimmune diseases, including autoinflammatory and immune deficiencies. Next slide. Regarding prevalence, this has been a significant issue over the years. NIH states that 24 million Americans have an autoimmune disease. This is constantly quoted in the press. However, this figure comes from a study that was done in 1997, actually sponsored by ARDA, which took a retrospective look at epidemiology studies of 23 diseases for which there were good epidemiology studies. NIH has not changed the figure since then, even though there is much evidence that autoimmune diseases have increased significantly over the past 20 years. The study was updated once by Glenda, who's present at this meeting, but the results did not change NIH's figure. So ARDA states that 50 million Americans have an autoimmune disease, which includes 153 autoimmune diseases. And this figure was derived from many sources and was a best effort attempt. So it was very difficult to collect this information, but we did the best that we could. Next slide, please. Recently, we looked at 18 autoimmune diseases and researched NIH sources for prevalence figures for those diseases. The results was a figure of 67,740,741. However, this figure would also include what I would say double dipping, because if a patient has more than one disease, they would show up in the prevalence in a paper on a particular disease. So the figure would be less than that. It's just that it includes patients with multiple autoimmune diseases. But nevertheless, we feel very confident that our 50 million figure is on mark. Just looking at these 18 figures and looking at NIH's sources, these were papers that in the NIH Library of Medicine. Next slide, please. So why is this important? Well, for policy decisions, research decisions, policy decisions are often made on the cost of autoimmune disease to society, or cost of diseases to society, the impact on society, help drive policy, health policy decisions, research decisions. Sometimes it's actually difficult to attract researchers to the category, to autoimmune disease research, because it is often difficult to raise funds to support the research, such as is possible in categories such as cancer and heart disease. Pharmaceutical investment. Prevalence also plays a part in what pharmaceutical companies choose to invest in. And in the case of autoimmune disease, most of the new therapies have come along in the last decade, have been pretty much centered around 7 or 8 autoimmune diseases, and then are used off-label in other autoimmune diseases. General public awareness and understanding is difficult when there is so much conflicting data and a lack of accurate data. While serving as president of ARDA, I was contacted by Netflix, CNN, Fox News, YouTube, and the Discovery Channel, all interested in doing a segment on autoimmune disease, which would have informed the public, but all dropped the story when there was so little accurate data and information to produce a good segment on autoimmune diseases. Next slide. Cost of autoimmune diseases. The cost of autoimmune diseases is completely unknown. In 2012, Dr. Fauci, director of the National Institute of Allergy and Infectious Diseases, estimated the cost of treating autoimmune diseases in the United States as greater than 100 million million annually. I'm sorry about that. No, actually it is 100 billion, I'm sorry. However, that figure has not been updated, nor actually has it been substantiated. If you try to find out the cost of autoimmune diseases, you are up against a wall. ARDA is funding a study on the cost of autoimmune diseases at the Autoimmunity Institute. The Autoimmunity Institute is a new institute in Pittsburgh that treats all autoimmune diseases. So their data, their collection is on all autoimmune diseases. But however, the study has been delayed due to COVID-19. So we have to wait for that study to start. Next slide, please. These are research areas that we feel need to have increased attention and funding. These issues were also identified by members of the Scientific Advisory Board. Research is very siloed in this country. It is getting better in that many of the treatments, particularly the biologics, are used in many different autoimmune diseases, from rheumatic to gastrointestinal and even neurology. However, still, research is pretty siloed. And this was brought up by our Scientific Advisory Board. They felt that the autoimmune centers of excellence was a good start in bringing about more collaborative research and crosstalk and research. But at NIH, they still felt it was pretty segmented into silos with not a lot of crosstalk. I'll skip down to the NIH because it was already mentioned, the Autoimmune Disease Coordinating Committee. This was a committee that was initiated by Congress many years ago. And the first year was actually funded with a $50 million budget. And that $50 million paid off very, very well and resulted in one of the research the study of families with multi autoimmune diseases, and created a registry, which was very exciting and really helped with research in the genetics of autoimmune disease. But from the first year on, after the first year, it's been an unfunded mandate. And at least from ARDA's perspective, and we do participate in it, it could be more robust, particularly if it had funding. There were some study groups that were formed out from that from the meetings, but still there could be so much more crosstalk on research if that committee were to receive increased funding. Even in the pharmaceuticals, there is siloing. I think in the future, one might think of a concept. Clinical trials are very expensive. There's over 130, 150, depending on which figure you want to pull out, autoimmune diseases. And we have found, particularly with the biologics, many of the treatments can overlap into the different disease specialties. So it might be something to at least consider how in the initial stages of clinical trials, there might be more inclusion of different autoimmune diseases, particularly if they're in the same specialty as rheumatology or gastroenterology or neurology, because they're very siloed, and then clinical studies have to be done on each one of the other autoimmune diseases that might benefit from that treatment. And we know in the biologics that's definitely been true. Next slide, please. So these are some of the areas that we've identified as needing more attention. I'll talk about fatigue first. According to an ARTIS study, which we had over 800 responses to a survey within 3 weeks, which that in itself is just amazing and shows what a problem it was that many patients would respond. So what we found with that was reported by a significant majority of patients across all autoimmune diseases. Fatigue was a factor for the reason for filing for disability, losing a job, divorce, and cited as the most difficult symptom to deal with. The majority of patients also reported that they did not believe their physicians took them seriously about this particular symptom. In fact, when a patient describes that we have coached them not to call it fatigue anymore, but to call it exhaustion, so that it might be taken up more by the physician as a serious symptom. But when you can see that the majority of patients are file for disability, it was because of fatigue, that's quite a significant finding. And so we looked through Medline for research into the causes of fatigue in autoimmune patients, and we found that there was virtually no research in this area, with the exception of one study in Australia on multiple sclerosis and fatigue. Okay, so that's also basic research needs to be increased, as much of the research in autoimmune disease is in single diseases, and the mechanism initiating autoimmune disease is still unknown. Infection and its role in triggering autoimmune disease is an important area of increased research. It has been long known that infection can trigger autoimmune disease, starting with rheumatic fever, but also including many other infections such as Epstein-Barr, hepatitis C, Helicobacter factor, and now recently, COVID-19 is triggering what appears to be, and I say appears because that's what's mentioned before, it hasn't been long enough to know if it's going to result in chronic autoimmune disease, but we do know many other infections have resulted not just in transient autoimmune, but in fact, chronic autoimmune diseases. This one is an interesting one, which Art is particularly interested in, and that's antiphospholipid. Antiphospholipid research happens to be in a silo all of its own, and there's just very little cross-fertilization when you talk to researchers in that area, especially in research in other autoimmune diseases. Most of the research with antiphospholipid is related to lupus, yet there is an abundance of studies that implicate antiphospholipid involvement in multiple autoimmune diseases, in gastroenterology, rheumatology, neurology, dermatology, and even ophthalmology. So antiphospholipid can also be present in infection, which sometimes is very transient, but as I said before, can lead to long-term chronic illness, and also in cancer. Therefore, it appears that antiphospholipid is a factor in the inflammatory immune response that would definitely benefit from much more research in that area. Epidemiology, I'll not go into further into that because it has already been discussed. Epigenetics, environmental triggers is a ripe area for further increased research funding, and its role in autoimmune disease. Genetic studies of identical twins has shown that no more than a 67% occurrence of the same autoimmune disease in both twins. Therefore, environmental factors must be playing a role in the development of autoimmune disease. research in autoimmune factors and its impact on one's genes is an area that could possibly lead to prevention through the study of epigenetics. But also, just recently, there was a study by NIEHS that by Dr. Fred Miller that showed that the presence of anti-nuclear antibodies in the American population has increased significantly in the past 20 years. And interestingly enough, it has increased particularly in males and young adult males who are not usually a typically thought of as a candidate for autoimmune disease. Now, that's not to say that they have autoimmune disease, but it does show the presence of anti-nuclear antibodies consistently in a person does often lead to the development of an autoimmune disease. So what that's telling us is we're going to have even more significant increase in autoimmune disease than we have in the past 20 years. So we'll look at genetics, and while there is much study, particularly in the MHC, that shows a significant role of genetics in autoimmune disease, and particularly in autoinflammatory disease, but clinically has not been useful information and eludes the clinician, especially in the area of diagnosis. So that's an area where if we want to get to earlier diagnosis, that is an area that might have some potential. The microbiome. The last decade has brought about much research into the microbiome and its role in the innate and acquired immunity. But one area that has not been subject to much research is the use of antibiotics early in life and the development of chronic autoimmune diseases later in life. There's some research, but it's only initial research, but very well worth looking into because possibly could lead to prevention. Next slide. This is continuing with the areas of increased research needs. So fatigue, I already mentioned that, and the basic research and infection. Next slide. Next slide, please. So antiphospholipid epidemiology, and now we'll move on to the last, the slide regarding patients' needs. So diagnosis has been a very significant problem for autoimmune patients. Not only is it very difficult for the patient, it's also very costly as patients go from doctor to doctor trying to find out what's the matter. We do a study also every 5 years to see if this is getting better. And what we find is the average patient sees 4 doctors over a period of 4 to 5 years before they actually get a diagnosis. So you can imagine the frustration for the patient, the frustration for the doctor, but also the cost of that. And again, I'm going to mention family history as being important, but it will not show up on any health history form that a patient fills out when seeing a doctor. And the patient does not know to share that information that perhaps their mother had Crohn's disease when they are having problems that are more likely to be arthritic. So physician education is important. We also did a study with over 1,000, we surveyed 1,000 physicians and found that the majority by far stated that they did not feel they received enough education about autoimmune diseases in medical school. The majority wanted more information, and a small portion, but nevertheless they were there, physicians who said because of the complications and the lack of knowledge, they would actually rather not see an autoimmune patient. So that's a problem for patients as well as for physicians. So physician education is very important and needs to be funded more robustly. Coordinated treatment. This is also a problem with patients. Patients have to see many different specialists. I see myself 5 different specialists. The last comment at my last visit from my neurologist is, this is not the way we should be practicing medicine, as she tries to coordinate information from all these different specialists. So that patients are very often left to coordinate their own health care. And it is very frustrating, and it is very expensive, and often results in sometimes over-treatment because the patient doesn't know they're getting the same type of medicine by two different specialists. There's also multiple, I think somebody made a mistake on that one when they were putting this together last night. There needs to be multiple specialist treatment centers, because that is where the patient could see different specialists at the same time. That is the model for the Autoimmunity Institute. The only other one that I know of in the world is in Israel, and they also have multi-specialists, and patients are gravitating to these centers. In the one in Pennsylvania, they have, in 1.5 years, they have had over 3,000 patients from all over the country come to their center to try to get a diagnosis. So maybe we could use an autoimmune specialist. Just put it out there as a thought, an idea. I know the other specialists might not entertain that as a good idea, but the patient certainly would. And then there needs to be more outreach to the minority community because of the problems of having difficulty getting diagnosis and access to treatments, and also the coordinated care. It's very difficult in that community to get coordinated care and access. Next slide, please. So these are the recommendations that we finalized. Research on computational biology of biomarkers to refine diagnosis on an individual basis. Epidemiology studies, data that will provide defensible basis to allocate research funding for the diagnosis and treatment of interrelated family of autoimmune diseases, and allow for an increased accuracy of health cost forecasting. Research regarding the immune system and the mechanism involved in the immune response, inflammatory response, and the microbiome. Supporting research resources and infrastructure by encouraging and supporting crosstalk among all scientific research with an emphasis on big data exchange and in the field of immunology. And I think that was mentioned before, the need for supporting research resources and infrastructure, particularly at NIH. Women's health research, because 75% of autoimmune diseases affect women, and one of the top 10 causes of death in women under 65. Just really needs more outreach to women and more research in this area. Increased basic research on the immune system, inflammation, and the microbiome would impact on autoimmune diseases, autoinflammatory diseases, infection, and cancer research. Next slide. Thank you for the opportunity to address this committee.
And thank you, Mrs. Ladd. I didn't think it possible when I saw that you were given a half hour to review the state of autoimmune disease in the United States to actually do that. But I think you did a wonderful job and informed the committee to help us with our work going forward. I have a very few minutes left for some questions. So if there are some from the committee members, I will entertain them for a few moments. Lisa, we'll start with you.
Yes, thank you very much for your presentation. I saw that most of the research priorities seem to be around basic research. Are there any aspects of research that focus on living with the disability that sometimes comes with autoimmune disease, and therapies such as rehabilitation therapies, ways to just allow people to live more effectively in their daily life?
Well, I think that research might relate to particularly the symptom reported by patients as being the most significant, adversely affecting their life, and that would be fatigue, which we are definitely encouraging. We have looked to PCORI. We have a grant submission to look at fatigue across big data to see how many times is it really actually being recorded in the families, I mean, in the patient's history, because patients really did not feel it was being taken seriously by their physician. And hopefully this would bring about more data that we can show the need for research into this particular symptom. As far as coping, we have actually added a psychologist to our scientific advisory board to help us with that. The impact of long-term chronic illness definitely takes its toll on one's life. So I would agree it needs more research, but there's very little research at this time.
Michael?
Hi there. I really enjoyed that, and you know that I agree with a lot of what you said. One of the issues, however, that you talk about when you talk about multi-specialty clinics and things like that is a restructuring of American medical care, American training, physician training, and so on. But this committee is dedicated to looking at what NIH can do. So in the very broad projects that you are suggesting, what should NIH's role be?
Well, NIH, which funds the Autoimmune Centers of Excellence, could increase those numbers. In fact, back in the beginning of those Centers of Excellence, they really only focused on research, and then they did begin to see patients and bring in clinicians as part of those. So I think that's a start where we could improve that coordinated care. And another possibility, and I'll just throw this out because we use telemedicine so much now, but I would just like to throw out an example that I think would work so well for autoimmune patients, and that is University of Mexico has a virtual pain clinic so that physicians all over Mexico, because as you know, pain can have separate categories, and many physicians don't know how to treat the different types of pain. But with a virtual clinic, they have all the different pain specialists there, the physician, the patient's physician, and the patient. They virtually have a meeting. They make recommendations, and then they follow that patient. So I think that type of model might work very well for an autoimmune patient. And then once the patient is identified with what disease they have, then they could see a specialist according to that particular disease with maybe ancillary specialists as needed. Just throwing it out there. A pipe dream maybe, but I think with telemedicine that we see working so well and the ability to meet virtually, I think that is a model that could be initiated by NIH in the Autoimmune Centers of Excellence.
So what I'm hearing is suggesting studies on different methods of delivering healthcare.
Yes, I mean, because it truly is left up to the patient to coordinate their care. And oftentimes they get opposing viewpoints from doctors on how it should be treated. And so it's left to the patient to make these very significant decisions that impact on their life, listening to 2 or 3 different professionals with different ideas of how it should be treated. This is just not the way, as my neurologist said, medicine should be done.
Very important point, really a very important point to end your discussion. And I'm sure we'll be having more discussions about that around the table. So thank you very, very much. Much appreciated.
Thank you.
We'll move on to autoimmune disease research in the United States, which will play up following this discussion of autoimmune disease. This will be done by Dr. Jane Buckner. Dr. Buckner is the president of Benaroya Research Institute, and that institute provides care for rheumatology patients at Virginia Mason Medical Center. She is an affiliate professor of medicine in the Division of Rheumatology at the University of Washington. Dr. Buckner, we're anxious to hear your comments.
All right, my video is about to get started. There,
There you
good.
go. There
Great.
you go.
Thanks so much for giving me the opportunity to talk about this gigantic topic. And I won't be able to cover everything, and I am going to be focusing a good bit on immunology research today, but I don't want to undervalue other types of research, but I thought I'd speak to what I am more expert in. Next slide. So just thinking about what the goals of your committee are, things that I'll be talking about today is thinking about how research impacts both our understanding of risk, diagnosis, treatments, and I think we can talk about prospects for cures and ultimately prevention. And I want to end by talking about some of the barriers that I see and gaps, as well as some really promising areas, because I think this is an exciting area that we're going to be able to make progress in over the next few years. Next slide. So as I started thinking about what I wanted to say today, I think it's really important as we approach the question of autoimmunity is how we think about autoimmunity really matters, because it impacts the way we ask questions. And so I think we have been somewhat over the years, less so today, but hindered by the fact that we frequently are searching for a single unifying mechanism for a disease. And I think that's really blinded us to the complexity of these diseases. And, you know, as a medical student, I was taught about Occam's razor, and I think it's very helpful in practice, although as a rheumatologist, it seems my patients seem to have more than one problem. And so I just thought this cartoon was helpful in thinking about how we frequently look for the simple answers, but they're wrong, and we really need to embrace complexity. And that's been a barrier, I think, to autoimmune research in the past. Next slide. We also have to acknowledge common features across autoimmune diseases while embracing heterogeneity within them. And so we always talk about lumpers and splitters. I think one of the features that has been difficult over the years, and it's already come out, is that we see each of our patients with autoimmunity sees a different specialist because they have a different end organ that's involved. And so for many years, people were studying all of these diseases individually. You know, we're now starting to study these diseases and what they share in common. And I think we need to go one next step, because I don't think they actually, as we look at these diseases, they don't actually lump as in an organized manner, but in fact, there's intersections in terms of the mechanisms that drive these diseases. So a disease, there may be the same pathway involved in lupus and type 1 diabetes and MS, and a different mechanism that links type 1 diabetes with rheumatoid arthritis, for example. So I think we need to be open to the concept of intersection, but not complete unity. I also think we need to embrace the fact that within diseases, heterogeneity is so important. And if we're going to move forward to understand diseases, impact appropriate therapy for patients, we have to really focus on that heterogeneity. I know there's a lot of people who study lupus on this panel, and certainly that's an area where heterogeneity is a huge impact on how we understand mechanisms of disease. And then I also wanted to bring out the idea that we shouldn't live in our own little bubble of autoimmunity. It seems like a big bubble, but in fact, we can learn so much from other disease groups. Thinking about cancer now that immunotherapy is being actively used, there's these links that I think are really powerful. Thinking about cardiovascular disease and fibrosis as disease areas that can teach us a good deal as we think about autoimmunity. Next slide. So the way I think about autoimmunity is that it's a failure of immune homeostasis and tolerance. And I wanted to put forth this concept of what happens to a patient as they develop autoimmunity. They really start as a healthy individual, develop immune dysregulation at some point, then tissue inflammation, so that immune dysregulation starts targeting a tissue or site. And then they'll get it at, that's where we start seeing the disease itself. But I also think there's a next stage, which is really disease perpetuation. And I think it's helpful for us to think about these as we start to understand how we want to diagnose people and also treat people. And we all know that genetic factors play a role at each step in the way, environmental factors. And I want to raise this point that was already brought up by Dr. Lashin, that time is a factor, and I think an important one as we think about the pathway to disease. Next slide. So it's already been brought up, and I know that this panel is well aware that we know genetic factors underlie the risk for autoimmunity, and we also know there's a growing literature about how epigenetic alterations also influence that. And I think this is really important in the setting of sex differences. And then these impact both immune function and fate, these variations, but also tissue-specific features. Environmental factors from exposure to infection, tissue injury, diet, exercise, stress, chemical exposures, all really important. And then again, we think about time, we think of growth, maturation, aging, and comorbidities that occur over time. We need to keep all of these in mind if we want to understand autoimmunity. Next slide. And so this is where I think we can start thinking about this model and think about how do we prevent each step along that way from health through autoimmunity development, clinical disease, and then this persistent chronic destructive disease. Obviously, our ultimate cure is to go all the way back to cure people, or certainly also, heal and improve people. And I think there's some opportunities that we haven't started taking advantage of. And that's when we can see reversion towards health, both spontaneously. We know this does happen in patients. Patients will be diagnosed with rheumatoid arthritis and spontaneously remit. People who have autoantibodies that are risk for type 1 diabetes, and they go away. So I think spontaneous, but also in interventions. I think if we can start studying the reversion back towards health, that would be ultimately incredibly powerful. Next slide. So I think a lot about how to induce tolerance in the setting of autoimmunity. And I think most importantly for autoimmune research, we need to know why tolerance is broken. And alas, I don't think autoimmunity quite falls into Leo Tolstoy's opening for Anna Karenina, because happy families may all be alike, but I think a healthy immune system, they're not all alike, and I'll show you some data related to that today, but they all achieve the same purpose. Whereas every unhappy family is unhappy in its own way. And I think we could say, well, autoimmune diseases share common mechanisms, but the complex interactions between these mechanisms and the tissues that they're attacking do make them unique in each way. So next slide. So thinking about what the roadblocks are to tolerance in autoimmunity, I think the good news is the mechanisms of failed tolerance are shared across autoimmune diseases. As I noted, genetic risk, immunologic functional features are shared, response to therapies are shared. And I think that's been the most enlightening thing in the last 20 years of my clinical practice, is to be able to use the same drug for multiple autoimmune diseases. The bad news is that the immune system is built to be redundant and complex. That's why it is so powerful in protecting us. That means multiple mechanisms are involved in retaining our tolerance, and modest alterations in several pathways in combination are actually the norm. So we get these complex multigenic diseases. And then ultimately autoimmune diseases arise over time. So the mechanisms that lead to failed tolerance may differ based on the stage of disease, whether it's at initiation, progression, or perpetuation. Giving us, I think, a more difficult road to really crack this nut. All right, next slide. So where have we fallen short in research? I think we focus too frequently on individual diseases or disease models. Yet we haven't started looking at target tissue that may drive disease. I think that's been a really highly ignored part. We immunologists love the adaptive immune response, but we really need to think about the organ it's attacking. We failed to embrace and tackle complexity. Simple or novel hypotheses are truly favored, and they are beautiful. Whenever I'm on an NIH review study section and I read those beautiful mouse models, I think, boy, that's beautiful research. And I think that can be powerful, but it has hindered some of the work in humans. So I think we need new approaches to deal with the complexity. We need models that reflect that complexity and analytical tools that help us tackle complexity. I think we failed to include the aspect of time in many of our studies. The majority of work that I've actually seen in autoimmunity are cross-sectional studies focused on established disease. Those are the patients that are easiest to recruit and study, certainly. I think natural history studies yield really novel insights into the mechanisms that drive each step of disease from initiation through perpetuation. So that is something I think is really important. I'm going to talk a little bit about that today. And then another failing is we lack an understanding of what immune health looks like. And as someone who's been studying patients with multiple different autoimmune diseases in my career, that healthy control cohort has been the one that has been most powerful for me and made me realize that heterogeneity within health is something we need to understand. And I think one of the things I'll get back to is responses to immune perturbations in health may help uncover causes for failed tolerance. So next slide. So what I thought I'd do, and I'll try not to go on too long, is talk about some successful research strategies and what they can teach us about how to move forward. So I'm going to talk a little bit about genetic associations that help us understand altered immune function in disease and the heterogeneity in response to therapy. I'm going to talk about mechanistic studies of samples from clinical trials, which I think enlighten us in understanding actually why people respond and actually underlying mechanisms of disease, and then briefly touch on studies of immune health and their implications in understanding autoimmunity. There's so much more I could talk about, but I won't be talking about some of the advances in understanding the fundamentals of the immune system through models. I don't want to understate how vitally important this work is. We have to understand how the car works before we're going to fix if it's broken. And those models help us do that. I think I'm not going to be able to talk about application of new tools to study the immune system. There are incredibly powerful new tools, particularly with gene editing. I don't want to undervalue those. And then also, it came up already, but expanding understanding of environmental factors, including the microbiome, is something that is an exciting area I won't be able to cover. Next slide. So I am going to talk about genetic risk, and of course, it's presumed to initiate environmental triggers to propagate autoimmunity. Next. So this has been demonstrated in lupus. Next. In rheumatoid arthritis, where we see this whole context of genetic risk, environmental factors, and development of autoimmunity until we get arthritis. And next. And the disease I'm going to talk about today is type 1 diabetes, because I think that's an area where our natural disease studies have been particularly powerful, and we have information about intervention studies. And I hope you are as amused as I am that I'm talking about type 1 diabetes when I'm actually a rheumatologist. Next slide. So genetic risks are shared across autoimmune diseases. I think we're all aware of this. This is just a brief demonstration of that for several different autoimmune diseases. And many of these genes are immunologic, which makes people like myself happy because we've been studying the immune system for so many years, but they're not exclusively immune genes, and some involve tissues. Now, the bad news about the shared genes is that typically for every disease, there's more than 100 genes associated with disease risk. Each gene confers only a very modest increased risk. So that the genetics of autoimmune disease are quite complex, and that already came out as I know there's concerns about how do we use that genetic information that we've worked so hard to get. And I hope I can convince you today that the genetics is leading us to understand mechanisms, and that may be where we can take this information and use it in patients. But one other part of the genetic risk, and that is that many of these genes confer traits that are fixed. That means we're born with that trait, and that means for us to impact disease, we have an individual who already has an altered immune system that may be hard to turn back. Next slide. So some of the ways we've used genetics, certainly rare gene mutations have helped define pathways important in autoimmunity. Example would be the IPEX syndrome, which is a deletion of FOXP3, which is required to make regulatory T cells. Individuals born with this disease have severe autoimmunity, actually leading to death by age 2. It includes diseases like type 1 diabetes, among others. But understanding this gene variant has led us to have a whole field of understanding how regulatory T cells are important in autoimmunity. Another set of genes, particularly the AIR mutation, leads to abnormal central tolerance, and APSED is the disease that patients who have the AIR mutation present with, which is multiple autoimmune diseases, all of them being predominantly these diseases are endocrinologic. I bring this up because, in fact, we were not aware that type 1 diabetes was an autoimmune disease until in the 1970s. Patients with APSED were studied and they were identified to have autoantibodies to islet cells. That really led us down the road to understand type 1 diabetes as an immune-driven disease. Next slide. So in that, knowing that autoantibodies are important in type 1 diabetes, I thought I'd share that following using natural history studies, and there's been a series of these studies done here in the US, supported by the NIH and others, as well as in Europe. But we've been able to establish over the years that actually in first-degree relatives in particular, having autoantibodies actually predicts development of disease. And this is a slide showing the age and years, the number of autoantibodies, and the probability of developing diabetes. And I would just have you look at 2 antibodies or greater. And we really get to the point where 100% of these individuals will develop type 1 diabetes. So we actually have a group of people who, with antibodies and important information about MHC genes, we can predict development of diabetes. We just don't know when. Next slide. So having information such as that antibodies are important in type 1 diabetes, interventions have been performed, and one of them is to show that depletion of B cells with rituximab, anti-CD20, actually was shown in new onset diabetes to delay beta cell loss. That's shown here on the left. The blue is the Rituxan treated, and we're looking at C-peptide area under the curve. All of these individuals had been recently diagnosed with type 1 diabetes, so they required insulin, but you're able to track the loss of beta cell function over time. And you can see that especially within the first 6 months after therapy with rituximab, the remaining beta cell function was preserved. Things that we learned about this is it wasn't permanent. We also learned that response was not uniform. And interestingly, among those who responded, the youngest subjects tended to be those who did the best. Next slide. So this is a little bit of the messy truth about the data in type 1 diabetes and the heterogeneity. And I'm using this, and any autoimmune disease is going to look like this. So we know that B cells are probably important in type 1 diabetes. On the left is a figure of the C-peptide area under the curve, but it's now for a very large cohort of individuals who were followed over time. So again, a study over time, and I drew in the red and blue lines myself, so I apologize, that wasn't in the publication. But you can actually define the loss. If we do linear regression modelings, we can actually define loss of C-peptide over time, and it falls into two groups. Those who are really slow progressors and those who are fast. Now, my colleagues here at BRI took samples from those subjects and did RNA-seq. And this is whole blood RNA-seq, and they were able to look at the profiles. And what you see in the fast progressors, who are the red on the left side of the middle figure, is that they actually have a B cell signature. And they were then able to show that the B cell gene sets were increased in younger age individuals. So rate of C-peptide loss is faster in the young. The B cell signatures are higher in young individuals. And there's this link between the B cell signature and C-peptide loss. And that brings us back to maybe understanding why young people did better with rituximab therapy, and maybe helps us think about who we would treat in the future, and indicates that B cells maybe are important in this disease. Next slide. So in addition to depleting B cells, genetic studies have expanded our understanding, and some of the common variants associated with type 1 diabetes promote B cell autoimmunity. The gene PTPN22 is a single nucleotide polymorphism that's actually the highest or most commonly shared risk variant across multiple diseases: lupus, type 1 diabetes, rheumatoid arthritis, for example. And that's our group showed that with others have shown that B cell tolerance is abnormal. Next. There's a variant in TIC2 that is protective for several autoimmune diseases, including type 1 diabetes and lupus. Next. And there are variants in genes that are involved in IL-2 signaling. These are associated with multiple autoimmune diseases as well and impact T cells. And I just wanted to put this picture here to show this intersection between B cells, T cells, the antibody production by plasma cells, and actually the innate immune response driving these responses. Next slide. So just quickly, we know that PTPN22 risk actually increases the amount of autoreactive B cells in the peripheral blood, and that's shown on the left. This is work from Eric Meffrey, who's now at Yale, showing that healthy controls who are CC, that's non-risk, compared to those who carry the risk variant, there's a huge increase in autoreactivity. And when we look at individuals with type 1 diabetes, we see autoreactive B cells across the board. So it suggests that there's failure in peripheral B cell tolerance in type 1 diabetes. Next slide. And I will just say you can extend these studies to RA and lupus, and you'll see a very similar picture. In IL-2, we were able to show that, in fact, if individuals who carried a risk variant for IL-2, if they were healthy, had an impaired response to IL-2 in their T cells. And then when we extended that, we looked in type 1 diabetes, and we also saw this failure to respond to IL-2, suggesting initially to us that it was a regulatory T cell defect. Next. But then some work done at King's College by Lucy Walker's group several years ago extended that work. They had identified in a mouse model that IL-2 actually impaired T follicular helper cells. T follicular helper cells are now the cells we understand are central to helping B cells as they create antibody responses. And Lucy's group was able to show that this enhanced the impaired IL-2 response led to increases in T follicular helper cells, and that was true in type 1 diabetics. Next slide. And the other gene that we also studied when we studied that TIC2 variant associated with type 1 diabetes and lupus is we saw that actually T follicular helper cells were increased in the individuals who carried the risk. Now on the left we have P, that's protective. So in red, people who have the protective gene don't have as many T follicular helper cells. They don't have as many switched memory B cells. So we know that this variant also has implications for T follicular helper cells. Next slide. So I thought I'd talk about this because this base of knowledge, understanding pathways that are altered in type 1 diabetes, has then led us to understand a little bit more about another therapeutic that we've used in type 1 diabetes, and that's a drug called CTLA-4 Ig, or abatacept, a drug that's used commonly in rheumatoid arthritis that blocks T-cell activation by blocking the CD28 pathway. And that's what I have figured here on the left. So many of us have thought about it as a block of stimulation. It was used in new onset diabetes, and that's shown down below. And the individuals in blue got drug, and those in red did not. And you can see that, again, we delayed the loss of beta cells in these new onset type 1 diabetics. So this is a drug we thought worked. We thought it worked because it blocked T-cell activation. But what Dr. Walker's group very recently was able to show, using samples from this same study done by TrialNet, an NIH-sponsored group, was that they could see that being given abatacept led to a drop in these T follicular helper cells in the blood of patients. That's what's shown. I'm sorry, it's a little bit cloudy. It's shown in the middle. And that when they started looking at the people who responded as compared to those who did not respond to the therapy, the responders started out with a lower number of T follicular helper cells. And that's what's shown here in the middle lower panel. This blue set of squares are the responders who really maintained their islet function throughout the course of therapy. And in fact, they were able to prove that they could predict response by measuring in the peripheral blood T follicular helper cells. So I think this is a really exciting understanding where we've come full circle. We know B cells and T cells are important. We have two therapies that target both of them. We're now at the point where we may be able to identify a biomarker that would have us identify the right therapy for the patients as we move forward. Next slide. So another study, and again, I'm focusing a bit on these studies in type 1 diabetes because we have wonderful natural history, and we also have interventions where we have been able to apply mechanistic studies. And this is a study done where I think the intervention has taught us something new about type 1 diabetes, and that's the use of teplizumab, anti-CD3 antibody. And it was shown quite a few years ago by Kevin Harold and others that when you treated new onset type 1 diabetes, that's in the top left corner in red, you preserve beta cell function, and in blue, placebo did not. And it's all falling off over time, and you're gonna say, boy, that looks like the last two graphs you showed me, but they're three different drugs. If you go below that, though, you can select a group of responders. They're in red, and you can see they really maintain their beta cell function throughout. Again, we looked at this using RNA-seq initially and saw that the individuals who were responders had this increase in a set of cells that were TIGIT positive, EOMS positive. Those are the blue dots on the volcano plot in the middle. We then took the samples and looked at them by flow cytometry, and the EOMS high people on the right had this increased number of the cell population. It's a CD8 T cell population, that's KLRG1, and TIGIT positive. This is an exhausted profile of CD8s. And below that, you can see over time the difference between responders, non-responders, and placebo in the number of these cells. And the black arrows indicate intervention and therapy. When this data first came out, we never had thought about the role of CD8 cells, particularly in response to a therapy, and certainly not this exhaustion profile. So it really raised our understanding of a new cell population in type 1 diabetes. Next slide. So the next point is that the goal is actually to prevent these diseases, and a study was done taking very high risk individuals who had 2 or more antibodies and dysglycemia. Next. Next. And that study was done in what we call stage 2. So the highest risk, but not yet diabetic individuals. And that study, and that's the wrong reference. That study was published in New England Journal last year and showed actually, next slide, that we can delay the onset of diabetes in this very high risk population. So these are individuals with type 1 diabetes was diagnosed in 43% of the teplizumab treated subjects, but in 72% of the placebo group. And when we took those samples back into the lab, we were able to show that the responders again had this increase in this CD8 population that was exhausted. This is really an important study because it shows that you can delay onset, not just slow the rate of progression, and gives us a new target for potentially preventing diseases like type 1 diabetes, but I think it extends beyond that to other autoimmune diseases. Next. So as I said before, I think if we want to get to cure, prevention, or reversal of disease, we really need to understand the target. And our target is to get to a healthy immune system. So understanding that. So next. And I just wanted to show some data that's kind of the type of data I've been producing throughout my career, where we observe a lot of heterogeneity in individuals, whatever parameter we look at. And here on the left, I'm just showing a response to a cytokine IL-6, and we have healthy controls in black and type 1 diabetics in red. And you can see that the type 1 diabetics have this higher level of response, and that's what we've been studying. But healthy controls are all over the map. So that's just one aspect. This is another on the right, another feature of CD8 T cells, and you can see the broad variation. So we see overlap between health and immune diseases when we look at individual pathways. Next slide. Now, if we start looking at people over time, and this is a study in diabetes, but we're doing it currently in healthy subjects, we see quite a bit of variation. So each of these numbers represents an individual who was sampled 10 times over 1 year. And this is looking at just one parameter, the CD8 effector memory. And you can see that each individual really is pretty consistent over time, but the individuals look quite different between each other. So I think understanding intra and inter-individual variation as we follow people over time could be of great interest. Next slide. And this is, as we think about people, diseases when they occur, we need to understand them in the context of the individual. And so studies now that I think are really important is understand what immune aging looks like and what's different about our immune system. And on the left, this is a very interesting study that took all of the characteristics from a study of the immune phenotyping using RNA-seq, but you can also do this with flow cytometry and other single cell approaches. And for every individual, they defined an immunotype, here shown in 3 dimensions, and each dot is a person. And you can see that over time, there's this change, and that change really represents aging. But they also took apart this data and looked at the age threshold for the groups. And in black, we have individuals who are CMV negative. In the circles, we have CMV negative, and then we have CMV positive in the triangles. And we see quite a difference because actually, if you've had CMV, which affects, depending on the population and location, between 40 and 70% of our population, your immune system actually looks significantly older. We've also recently been studying Down syndrome and using an algorithm that predicts immune age. I've seen that their immune age is 20 years older than an age-matched individual, even as children. So that, and they're at very high risk for autoimmunity. So I think immune aging is something we need to understand and understand some of the factors that influence it. Next. Other things we need to understand is seasonal variation as a source of immune heterogeneity. I think this is a beautiful paper done by the group in Oxford, John Todd's group, showing when they did just RNA-seq on the immune cells of the blood, we see this seasonal variation. There's summer genes in blue and winter genes in green. And so we need to start thinking about that. Certainly autoimmunity can be seasonal in terms of when people see flares or when they see new onset. Next. And then diurnal variation, other lifestyle impacts, and this is a study, again, I think is quite interesting, looking at sleep. where we can, on the left, just look at the frequency of CD4 and CD8 T cells varying over the time of day that you draw the blood. And as we study human beings, we need to keep that in mind, that if the blood is drawn at 7 a.m. and 8 p.m., it may look different. And then one of my favorite studies is this vaccine study where a group of male volunteers were vaccinated. They were either allowed to sleep the night after the vaccine, or they were kept awake all night, and their response as measured by their antibodies was done 21 days out. And you can see having a good night's rest gives you a much better response than not. And this is from one day. I think it's pretty exciting data, and I will be getting a good night's rest after I get my COVID vaccine. Next slide. Other lifestyle sources certainly lead to immune heterogeneity. Those of us who follow the rheumatoid arthritis studies know that smoking doubles your risk of RA in the appropriate genetic background. So understanding what these types of lifestyle factors can do to our immune system, I think, is really important, and there's growing understanding of that. Next. And then ultimately, I just wanted to show you some very exciting work people are doing. This is complicated stuff, but there's been some really lovely work using twin cohorts where we're able to dissect what's genetic and what isn't. And here on the left, we can see a characteristic of CD4 T cells where you look on one axis it's comparing the twins, and you can see that this CD4 population is very much genetically driven because monozygotic twins look almost identical, whereas dizygotic twins are quite different. So I think this helps us get at the gene environment question in a beautiful way. Next. And then there are groups now collecting large cohorts of healthy subjects using all of the incredible tools we have, both by single cell cytometry, RNA-seq, ATAC-seq, for example, to understand the genetics, epigenetics, as factors that drive the variation within healthy human beings. Next slide. And then I haven't talked much about models, but one of the things I'm going to say is we need better murine models to imitate human biology and to even test therapeutics. And I think I was always surprised when I realized that my adult humans didn't look anything like my colleagues' murine data. And that was because they were studying babies that had no exposure to any pathogens or even microbiota. So I think some of the beautiful work that's now being done with the pet store mice, showing that you can actually have animals that are co-housed with different microbiomes really look much more like mature human beings. And I think that's kind of a revolution in our thinking about how to develop models moving forward. That does mean it's more, you have more, unfortunately, and what the figure on the right shows is that once you start making mice more like people, they look more like people, which means there's more heterogeneity. You need to study more animals if you actually want to get to the bottom of things. So it comes with some challenges, but I think they might be important challenges for us to accept to move forward. Next. So what I've been talking about today is really a vision of thinking about autoimmunity in a way that I call a well-orchestrated deep dive. You know, certainly basic research is important, but then taking human samples, particularly well-characterized human samples where we study over time and bring them into the laboratory and study their immune characteristics, their genetic characteristics. But I also think that applying that to our patients in terms of helping select therapy, and then when we do an intervention trial, to go back to the lab and understand why it works, why it doesn't work in some people, so we can do better. Next slide. So I just thought at the end of this, I'd talk about some of the barriers that I think exist in terms of NIH-funded research. I think a lot of these projects require development and maintenance of infrastructure over time, especially longitudinal studies. And that's something that we really need to have the long view on. And I'm afraid that if your grant's every 3 to 5 years, that can be hard to do. So something to think about, how do we do that? Another area is large consortia. They can be excellent, and there's some really nice examples of that, but they can also be ungainly and hard to administer. At least that's been my experience in some settings. So I think, as well as thinking about how do we do these projects, sometimes really small committed groups can prove to be really productive. And I think looking for those can be helpful. Human immunology has really increased challenges to demonstrate productivity and to get high impact publications. I think that's improving because many of our major journals are actually looking for human studies. But I would say for the last 20 years of my career, this has been a challenge for myself and for others. And I will tell you, it really decreases the desire of junior investigators to pursue this path, which I think is important for us to move forward. And then the other challenge is for computational biologists. This has already been brought up. I think they're really going to be vital to helping us deal with complexity, but the pathway for them to be recognized as independent investigators is difficult because, in fact, they have to work collaboratively with people who are collecting samples, studying, generating data. So we need to have them encourage collaborations and figure out ways to help recognize them and support them. Next slide. Some potential pitfalls. I said too big or too small. I think sometimes I look at big data and it's too big because I look at the papers and I go, okay, that was kind of cool, but what does it mean in terms of patients? So I do think we need to not only look for big data, but look for how do we take big data and make it meaningful for patients. Too small is if we only study one model forever and hope that that's really going to describe the disease our patients have. And there's already been quite a few papers about this. We've cured the NOD mouse hundreds of times from type 1 diabetes. I think it's actually a really good model, but we have to move beyond that one model or these very targeted, limited models where you only look at one pathway. And then I don't think we should be blinded by shiny new technologies. I don't think innovation should be the point, but it should be a way to get to the answer. So I think we should look at what new technologies bring us to answer the questions we have and integrate them with tried and true methods that have been developed. And I think we have to just think seriously about that as we move forward. Next. Oh yeah.
I think we have to curtail towards the end, so we can allow some time for the committee to give some questions.
Yeah, this is actually my last slide.
Oh, good.
I know, I was the, yeah, so this is really my last slide. And what I thought is some of the gaps, and it's really pretty, it's review. It's develop and improve models for human disease, embrace heterogeneity and the disease mechanisms and interventions, and expand studies over course of time. And you know, I've emphasized that we need to define health and expand inclusion of environment, lifestyle, demographics as variables that influence health and disease. So I can end there.
Well, thank you very much, Dr. Buckner. This has been a very comprehensive view of autoimmune disease research in the United States and very helpful to the committee as we begin our deliberations. I'm sure we'll have questions, perhaps not time for today, but we will have questions in the future that we will ask, and perhaps you'll be kind to give us information that we can work on as we go forward with the committee. We do have some time for questions, and if committee members do have questions for Dr. Buckner, we would be happy to entertain them, so please raise your hand. But at the same time, I think it would be a good idea that if we can entertain a few questions from the audience on the Q&A while we're entertaining questions also from the committee, that would be helpful. So perhaps Dr. Buckner can answer those questions in addition. So I'm going to open up right now. We do have a few minutes to do both. And once again, Dr. Buckner, thank you very, very much.
So, well, I can get started with one of the Q&A questions that came in from the participants. A more general question, big picture question is has to do with why there is such a huge epidemic of autoimmune diseases. I think some of the presentations have touched on the different pathways and the different drivers, but I was wondering if perhaps Dr. Buckner could take a shot at answering that. And if Ms. Ladd is also on the call, and one is still with us and wants to answer that too, that would be great.
Yeah, I mean, I have a comment. I can make a few comments. I don't think we know why there is, and I think it's a really important point that there are increasing autoimmunity as we follow many different diseases. I think that's an important area for us to investigate. Again, I think that likely points to environmental factors and lifestyle factors. And that means to understand that we need to not look at the time you've got the disease, but before you get it, and understand also people like myself who are studying immune features need to partner with people in epidemiology who are studying the features of the environmental factors that may be driving this.
Michael?
That was a wonderful talk. One of the things that is a problem in translation from the mouse to the human and vice versa is that clinical research generally requires very specific phenotypes. And the point you just brought up is something that's bugged me for a long while, is that you've got a 10-year lag between onset of immunologic abnormalities and development of lupus, but all those patients who are participate in clinical trials, for instance, or clinical studies have passed that period and they're not eligible. Do you have a way of dealing with the phenotype specificity versus the ambiguities when you try to translate between mice and humans?
You know, I think, I'm not quite sure in terms of the question, is it how this difference between the fact we can watch a mouse over time, but we can't, the human walks in and they have the disease. I think one, we need to start thinking about how to study human diseases where we take it back, which takes a lot of effort. And then in mice, I think we need to look at aged mice more, and we need to develop models that will allow us to look at that development of disease in models that bring in some other environmental factors. That's some of my thoughts around that. I'm sure there's many other approaches.
Well, any other questions from the members who are?
Yes, I think this is a clarifying question on Dr. Bachner's presentation. Are autoantibodies in type 1 biomarkers the only, or are they one of the causes for the destruction of beta cells?
It's a terrific question. So actually, I did emphasize the antibodies, but in fact, we believe that the destruction of the beta cell is likely due to T cell infiltration of the islets. B cells are important in driving the autoimmunity. We're just getting an understanding of the role of each of these cell types, as well as their interaction with the islets and what may be happening in the islet itself to drive autoimmunity. So there's not one single thing. If we had that, if we knew it was only one antibody, I'd have a much better way to actually treat the disease. So we think there are multiple pathways that get you there, and we need to think broadly about that as we go into therapies.
Jill?
Hi, Jane. Thank you for the fabulous talk. My question is really more about some of our shared experiences. You mentioned, and I'm fascinated by the combination of autoimmune diseases and the common etiologies and mechanisms. But I think we also know how difficult it is to study that in humans with the Signature Project, et cetera. But do you have any idea, do you think that one of the barriers at NIH is the fact that there's multiple institutes involved, and getting a funding mechanism of some sort might be more difficult, particularly given that long-term structure that you mentioned to get some of these natural history studies underway. Do you have any thoughts on what might help foster some sort of cross autoimmune disease research?
Yeah, and I thanks for bringing that up, Jill. And we know, Jill and I both know how hard some of this work is. you know, one of the things I run into is the silo question, and it's not just at NIH, it's very true in pharma. You heard me talk about type 1 diabetes today. I frequently talk to people who know nothing about the type 1 diabetes world and are trying to figure out RA. And I think maybe thinking about structures where we say we want to understand natural history of autoimmune diseases and develop an approach that will bring in multiple different autoimmune diseases. It doesn't have to be the same group doing all autoimmune diseases, but I think bringing those groups together so it's a little more coordinated. I think that would be really powerful. And you're right, because we have, you know, we get funding from multiple different groups, they aren't all talking to each other. And I think we would really benefit from those conversations and shared projects.
Thank you.
I have two more people in the queue, Deirdre and Andrea. So let's take one quick question from each to end the session. Thank you very much, Andrea.
Sure. Hi, Jane. That was a really wonderful talk. My question is very much related to Jill's question, actually. It's very fascinating aspects of type 1 diabetes research that you presented. And being a rheumatologist, you can probably understand that, you know, some of our diseases, they don't have that many patients. The dynamic might be different in terms of doing human research. So what do you see as like particular challenges for autoimmune diseases that are not maybe as common as type 1 diabetes and in that light?
Yeah, so I think you're absolutely right. So, you know, I think places where we've made more progress is actually in some of the pediatric autoimmune diseases. So pediatric lupus is an area where there's been some really beautiful work done. And I think when you talk about the pediatric diseases, you kind of are enriched more genetically for risk. I think what would be an approach that we could use is first have really well curated early disease studies so that we can look really early in disease. That takes a lot of work from the point of clinicians. You're telling someone they have a diagnosis of a chronic lifelong disease, and you're asking them for a blood sample or participation in however you want to do it. But I think we should push as early as we can to start looking at patients. And that's going to give us, and then follow them over time, because I'm also, as a rheumatologist, I didn't talk about this, but I actually think this persistence of disease and difficulty treating patients over time is a huge problem. And we need to address that. I you know, have been seeing the same patients for 20 years, and I'm out of options for some of them. So I didn't talk about that today, but I do think longitudinal studies, not just before disease, but actually during disease, could help us, because I think there's stages even once we've diagnosed them.
Thank you.
Thank you. And lastly, Deirdre.
So thank you for that wonderful talk. So very briefly, and I think you may have covered this, are there existing cohort studies of any, you know, NIH funded cohort studies of any of the more common autoimmune conditions? And I'm asking this question to just build upon a point that you were making about the challenges in terms of junior investigators pursuing autoimmune diseases as a discipline, that they may not see the productivity from it because it takes so long to kind of have data to work from.
Yeah, that's exactly, is my point, that once you start getting data, sometimes it's 10 years down the road, but then it's a really rich data set. So there are some lovely cohorts. You know, I've talked about the type 1 diabetes, and they have been doing that through TrialNet, and before that there are others, and there are other groups that have cohorts, like the DAISY cohort in Denver. There are lupus cohorts that have been developed that are quite excellent. Judith James group in Oklahoma is one, but there are others. I don't want to miss, there's many people doing this. There are at-risk RA groups being done. Jill knows that group well in Denver, and I collaborate with them. So there are groups doing this, and there are more, I'm sure. I'm not listing all of them. One thing I can say is, if you look to the Europeans, they are doing this much better than we are. So we actually have a model if we want to look there. And I think that we should be able to do as well, if not better. But the EU has done a really good job of making this an emphasis, and they have some really, in rheumatoid arthritis, particularly outstanding work.
Well, thank you very much once again. And let me give a big thank you, big shout out to our speakers, Dr. Gomons, Mr. Keenan, Ms. Ladd, Dr. Buckner, and Dr. McNamara. And thank you all who tuned in today. Sorry we didn't have time to give a lot of receive a lot of questions from those who tuned in, but I do believe if you would send those comments or questions to Henrietta Awo OCI Anto, the project study director at autoimmune@nas.edu. That's autoimmune study@nas.edu, autoimmune study@nas.edu. We will try to answer those questions. But please keep in mind that any material you wish to share with the committee will be made public through the project public access file. That is because the National Academies comply with Section 15 of the Federal Advisory Committee Act rules. I believe I do speak for the entire committee when I say this has been a very informative session, and we are definitely eager to continue our work. I think the committee can take a well-deserved break for 15 minutes because we will reconvene after a 15-minute break at approximately 4:15 or 4:20. Thank you very much all.